Hello, and thank you for taking the time out of your day to join this virtual conference. What I'd like to do today is tell you about the leap-in transposase platform that we have developed at ATUM and how we've applied this to the development of stable cell lines for the manufacture of protein biologics. Cell line development, as you're all aware of, is a relatively long and tedious process, which begins with the design of a vector, the synthesis of that vector, and of course the transfection into the CHO cells. Stable pools are generated and ranked, and typically this ranking is done on the base of titer. Following pool ranking, a subset of the pools are then subject to single-cell cloning and the screening of thousands of clones to identify not only high-producing clones, but also those producing product with the desired product quality attributes. Of course, the clones generated must also be stable. Then a research cell bank must be manufactured and released to the GMP environment. Although timelines vary, I think we all would agree this is a very tedious and labor-intensive process. Before I talk more about how our platform has addressed these issues, let's talk about the life of a transposon and a transposase in nature. Transposons are genetic elements in DNA wherein inverted terminal repeats flank the transposase gene. This has evolved over many years through evolution, and the goal of this genetic element is to move from one place to the next, creating genetic diversity. It does this via the expression of the transposase gene, which is harbored within the transposon. Once expressed, the transposase enzyme recognizes the cognate ITRs and moves the transposon from the initial location into the new chromosomal location. In this case, it's identified by the tetranucleotide motif, TTAA, which you'll also see repeated here on either side of the inverted terminal repeats. Importantly, this is a cut and paste mechanism where the entire transposon is moved from one location to the next. The initial location is sealed up in a scarless fashion, and a single copy is then integrated at the new site. Importantly, there is a perfect high-fidelity integration of the genetic elements between the ITRs. Many of you have probably heard about this system, and not the least of which is via Barbara McClintock, her work in maize, leading to the 1983 Nobel Prize. So how do we apply this to biotechnology and creating CHO cell lines? I think it's quite evident to most of those listening to this. The transposon is, of course, integrated into an expression vector, wherein the gene of interest and all the regulatory elements are contained within the transposon on a circular DNA plasmid. You'll notice the absence of a transposase enzyme. This is provided in trans as a transient co-transfection of the mRNA along with the DNA plasmid. Once the enzyme is expressed, the transposition event occurs. The transposon is now relocated to the chromosomal target site. The remaining plasmid is recircularized and degraded inside the cell. This is a stable event because the transposase enzyme is only expressed transiently. A single copy is integrated at each site. There are multiple integrations across the genome. And importantly, and I'll spend most of the talk today focusing on this, the structural integrity of the transposon is maintained. That is, what has been designed here in the expression cassette will in fact move from the DNA plasmid as is into the chromosomal target site. And of interest, there seems to be no size limitation to the transposon that can be moved from one place to the other. So what that means is the result is a consistent and uniform integration of these leap-in transgenes. And what do I mean about that? Let's say we have an antibody with a heavy chain and a light chain that needs to be expressed, and in this case, a GS selection cassette for use in a GS null cell line. If you're doing random integration, of course, there are occasions where you may get what you've designed, but the reality is much more complex. There are truncations and deletions, as well as random concatenamers and scrambling. Therefore, what you've designed in silico, what you've built in your vector, is not necessarily what ends up in the genome. And in fact, we know that this is the case. Contrast that to the transposase system, wherein you put ITRs on either side of your expression cassettes, add transposase, and you get perfect integration of what you've designed and built into the host cell genome. Importantly, the functionality of each one of these insertions, on average, is comparable to each other. And this is true not only of the expression level, but also the product quality. What this does is create pools that are highly uniform and quite predictive of the clones that can be derived therefrom. So what can you do with these pools? If you could design in silico and be 100% confident that your design will end up in the genome, how might this be used to solve problems in expression? Well, I'll give you a couple examples of that. In this case, in all of these cases, we'll be talking about the controlling of different chain ratios in multi-open reading frame molecules of interest. In this case, I'm going to talk about a 2-orf molecule. You can think of this as a bispecific in a format shown here, or you could even just think of this as a heavy and light chain antibody, wherein perhaps you need excess light chain compared to heavy chain, or not. In any case, in this experiment, you see 14 different constructs that have been generated, where we have designed them to have varying ratios of one open reading frame to the next. Construct number 1 having a large amount of the pink chain compared to the blue, and construct number 14, you can see that that has been flipped. Again, these are all generated rapidly in pools and then screened for the relative ratio of each chain. This can be done with more complex molecules. There's a 3 open reading frame situation. Again, you can think about it as a knob into hole bispecific in this format, or any other 3-chain format that you may be using. In this case, 6 different vectors were designed, and you can see the relative ratios of the expressed open reading frames. So why does this matter, and how can it make a difference? I'll show you a couple data from one of our licensees, where they are in fact developing 3-chain bispecifics. And you may be aware that in doing so, oftentimes the desired product is not always what comes out. In many cases, there are many assembly variants, and it's thought that the relative ratio of the individual chains could drive proper assembly of the product of interest. And I'll show you a couple of examples of that. Again, this is data generated from one of our licensees, where in this particular program, their in-house system gave them approximately 550 milligrams per liter of this complex bispecific. However, only about 350 mg per liter was the actual molecule of interest. Contrast that with a leap-in derived pool wherein the chain ratios had been optimized, and you can see a gram per liter of total product, but more importantly, 900 mg per liter of the desired product. A massive increase of over 3-fold tighter in the desired product of interest. And in this example, I find quite striking the in-house platform actually creating quite a bit of protein material. However, the vast majority is not the desired product, only about 100 to 150 mg per liter of the properly assembled molecule. Contrast that with the leap-in derived pool, optimized chain ratios in excess of 1.1 gram per liter of the desired product of interest, a 10x improvement in desired product titer. And how do So at this point, I'd like to give you a summary of what I've told you so far. The leap-in transposase system creates multiple insertion sites in the genome. Each site is a single copy. I've not talked about it, but the enzyme prefers open chromatin. And importantly, for each insertion, the structural integrity is maintained. On average, each integration is functionally equal. And because there's no size limitation, we can build relatively large and complex vectors. How do we create these pools? These pools are created in a drug-free manner. We work with the Horizon Discovery GS knockout host for the majority of our R&D and platform development. And in this case, it's a drug-free selection simply by removing glutamine, but we can in fact vary the stringency by using regulatory elements within the vector. Importantly, we get rapid selection in less than 2 weeks and reasonably high titers. In this case, you can see selection in about 10 to 12 days and titers in the 2.5 to 3 gram per liter range. I'd like to mention that our next generation vectors actually select even quicker than this. We see selection in about 5 to 7 days and even higher titers. Here you can see an example of 6 antibodies showing titers from 5 to nearly 6 grams per liter, and in this case an Fc fusion molecule and another 3-chain bispecific in the 3 to 3.5 gram per liter range. So what is it that's so unique about these pools that allow us to achieve such high titers and such unique product quality attributes? And the reason is that the population of the clones within these pools is very uniform and in fact shifted towards high-producing clones. In this example, if we analyze clones derived from a pool, we can see that 62% of them are in fact in the top quartile of expression, with 82% in the top half of expressors. If we compare this to random integration, using some data and statistics taken from the Berkeley Lights website, random integration results in a distribution that's much more skewed towards the lower expression ranges, and the high expressing clones are actually quite rare, suggesting that at least 5,000 clones would need to be screened to get a 99% probability of finding these rare high expressing clones. Using the same statistics, we can identify same clones in under 200. So what does that screening look like? We use the Solentum VIPS instrument to help us with this. In this example, we screened 166 clones in static 96 well plates. We took the top 22 to deep well 24 well format in a 7-day fed batch. The top 8 clones were chosen for shake flask studies. Not surprisingly, given the uniformity in the selection process, the top 8 clones are relatively similar in their expression levels, in this case between 3.5 and 4 grams per liter in this simple shake flask format. One of these clones was then taken and done a very simple media and feed analysis, and very rapidly was able to find a condition in the 6, nearly 7 gram per liter range. To be clear, this was not a process development program. This was one experiment where a new media and feed scenario was tested. This is not only true of our internal programs, but also our external programs, where we see the stable pools actually being very predictive of clonal titers. Here's an example from Janssen, one of our licensees. In this case, the shake flask titer of their pool was about 4.5 grams per liter. And as predicted, the shake flask titer of their clones, again in the 4 to 4.5 gram per liter range. Their scale down model for their phase 1 process happens to be an AMBER 250, and those same 8 clones in an AMBER 250 yielded the following results. Not surprising, some clones behave better in the AMBER 250 than others. However, none were any worse than what we saw in the shake flask. And in this case, we see a 4.5 gram per liter pool yielding nearly 8 grams per liter in the AMBER 250. The same is true here for a molecule that's not as high expressing, where the shake flask in the pool yielded approximately 3 grams per liter, was very predictive of the clones in the same expression format, and in the scaled down model here, multiple clones in the 6 gram per liter range. But what I'd like you to take home is that not only are these pools predictive in terms of the titers that they can generate, but also the product quality. In this case, we're looking at glycan profile from pools as well as clones, and you can see highly similar glycan profiles between the pools and the clones, as well as the charge variance. Again, pools and 5 clones derived therefrom. None of this would be useful if these clones were not stable. I assure you this platform is extremely stable. Recall, The transposase enzyme that catalyzes the transposition of the transposon into the genome is only expressed transiently, and therefore it is gone within 48 hours of transfection, and hence the mechanism for excising the transposon is no longer active inside the CHO cells. In this experiment, we have 9 clones where we've looked at their stability for 60 days in the absence and the presence of glutamine, and compared the copy number at time 0 in gray with those other 2 conditions in the pink and the blue. And what you can see here is for all 9 clones, the copy number remained stable. We see an average copy number of about 25 to 30 copies per cell, but you can see ranging from in the 20 to 40 range. The expression tells us the same story. Again, 9 clones, absence and presence of glutamine compared to the beginning of the study. And what you see here is that 8 of the 9 clones had 100% retention of expression. And here, clone number 5, a slight decrease after 60 generations, but only in the presence of glutamine. You'll also notice that clone 5 retained its copy number, and so this reduction is not due to a loss of gene copy number or genetic instability, but rather appears to be some localized silencing. If we look at this over hundreds of clones, the following statistics start to fall out. Over 200 to 300 clones that we've analyzed, if we look at copy number at PD60, in the presence of glutamine, we can see that over 90% of clones retain 100% of the copy number, and this exact same story for the expression. Over 90% of clones retain 100% of expression after 60 generations in the presence of glutamine. And what this means is that genetic stability is no longer on the critical path. It is not a clone ranking parameter. You can move forward with tremendous confidence knowing that even if you only choose 3 or 4 final clones, they are highly likely to be perfectly stable. So in summary, we've taken this long and tedious process and we've significantly shortened it and made it more robust. The current process gives us approximately 10 to 12 weeks from transfection to the cell bank. In 3 to 5 weeks, we see representative pools being generated with clones available approximately 2 months later. And as you can see, genetic stability studies overlapping with our final clone ranking efforts. Summarizing the power of these pools and why they're uniquely enabling. Recall that the pools predict the clones, and this is true not only of titer, but also many charge variant characteristics and product quality characteristics. This enables one to screen vector configurations at the pool stage and then choose the appropriate pools for cloning, which can be uniquely enabling for more complex molecules, and more importantly, enables the rapid and robust progress towards IND. Not only by early transfer to a CDMO of pools and pool-derived material, which can enable process and analytical developments in parallel with clone selection, but we're talking to many groups about the possibility that these pools might in fact be used for tox lot generation as well as phase 1 manufacturing. If you may be aware, this is a relatively new technology launched just a few years back. We offer this as a service and have delivered on over 20 projects to date for our clients. We also license this technology and have licensed over 25 different companies now, and you can see 10 of the top 20 pharma. There has been an IND filed and accepted by the US FDA, and this was done in 2019, and we expect at least 3 more this year. You can see this is very rapid progress and uptake for a technology that is only about 2.5 years on the market. So with that, I will thank you for your time. And again, hope that you've learned something today about the LeapIn platform, how it can reduce timelines and robustly generate high titer clones, which may in fact be enabling for the next generation of biologics. So with that, I will thank you for your time and give recognition to both Solentum and Horizon for very fruitful collaborations we've had over the years. Thank you so much, and I look forward to answering any questions you may have, and in the not too distant future, hopefully seeing you all in person. Thank you.